So there's generally not a lot of drama or big scandals in the world of science, but this woman here, Rosalind Franklin, is actually at the center of one of the big controversies in DNA science. What's this about? Why is she such an interesting figure, and why is she at the center of a big controversy in the world of science? In this video, when we talk about DNA, we'll get to the bottom of Rosalind Franklin and her contributions to science. Let's get started. So imagine a time when we didn't have an idea what DNA was. We take it for granted today that DNA is what makes us who we are. We know it's a double helix. We know all about DNA. We even modify it and manipulate it all the time. But there was a time when we didn't understand what DNA was, how it worked, what its structure was. So there was a time in history that there were a lot of really important scientific historical experiments that were going on that led to our understanding of DNA. So the proof that DNA was the carrier of genetic information, that was a really important concept that we didn't understand several decades ago. So what we're going to do in this video at the beginning is I'm going to walk you through several experiments. These folks here all contributed to this discovery of DNA. Griffith, Chase, Rosalind Franklin, Watson, and Crick. And what we're going to do is I'm going to go through what each of these experiments did, and then we're going to talk about DNA as we understand it today. So Frederick Griffith sort of started the whole thing, and he did some bacterial transformation in mice, which he called something a transforming factor. Then we're going to talk about Hershey and Chase, their work with bacteriophages. Rosalind Franklin, who I started the whole video with, her work with X-ray crystallography. And then finally Watson and Crick, who proposed the double helix model of DNA. So at the beginning of this video, we're going to go over these 4 specific contributions to the discovery of DNA. So let's start with Fred Griffith. There he is right there. He started this whole thing by doing some experiments with mice and bacteria. So we knew that there was a streptococcus or a Streptococcus pneumoniae bacteria that was pretty pathogenic, right? So he did some tests. He took some of that living strep bacteria, injected it into the mouse, and as we suspect, the mouse died. Then he took a different strain of that Streptococcus, the R variety, which we knew was non-pathogenic, and he injected that into the mice. Guess what? The mouse lived. Well, this was a non-pathogenic bacteria. We know that good bacteria can live in our body. This one particularly lived. And so then he took the bad strain, the S strain, and then heat killed it, right? Same thing we do to our food. Kill it with some heat so all those bacteria die. So heat killed it, injected it. Guess what? Mouse lives, right? Then in the interesting experiment, what he did was he mixed these two. So the living strep R and the heat killed S. Neither one of these, notice, killed the mouse on their own. But notice what happened when he mixed them together. The mouse died. That didn't make much sense. Before, neither of them on their own killed the mouse, but when he mixed them, it did. Well, what we now understand is that he was doing the first, what we call bacterial transformation experiment. And what happened is, we now know that bacteria can transmit DNA to each other. So some of the bad genetic material from this heat-killed pathogenic bacteria actually transferred to the good bacteria, and that actually ended up killing the mouse. So he didn't know what this was called. He called it the transforming factor. We now, of course, know that that's DNA that moves between the bacteria. So then the next example of some work that led to our discovery of DNA was Hershey and Chase. There they are there. And they did some experiments with bacteriophages. So a bacteriophage is basically a virus, right? There it is right there. There's the virus that infects bacteria. So it's a phage, a virus that infects bacteria. And what they did is they used some radioactive labels. So What they did is they had these viruses that grew in different media, and they labeled the phosphorus with some radioactive in the DNA radioactively, and then the protein, which has a lot of sulfur, they radioactively labeled that. So in this first example right here, what you see is here's the bacteriophage, and inside is that radioactive DNA, because DNA has phosphorus and that was radioactive. Then they labeled the protein coat. It's kind of hard to see, but this outer protein coat here on the virus, that had a lot of radioactive sulfur. And what the point of this experiment was what gets transferred into these bacteria? Is it the stuff with the phosphorus, which we now know is DNA, or is it the stuff with this protein? What's doing the damage? Is it this protein on this virus, or is it this stuff inside? Well, what we found out was that actually, for this experiment with the radioactive phosphorus, we found the radioactivity inside the bacterial cells afterwards. So that said, what that meant was something that was inside the virus, we now know as DNA, was actually transmitted into this cell. But over here, in this experiment with the radioactive sulfur, there was no radioactive material found in the bacteria. So that means the radioactive stuff was actually the DNA. Okay? So this was one more step in discovering what it is, what's this transforming material, what is it that passes these genes, this heredity, on to other things. Then Rosalind Franklin, the source of the controversy we talked about. Rosalind Franklin was a brilliant scientist, and she worked in X-ray crystallography. And her work actually led to some basic helical structure. She suggested DNA had this sort of helix shape, and even this sugar phosphate backbone in DNA, which we now know is true. So she actually had a lot of the really, really fundamental ideas of DNA down. In fact, photo 51, this famous photo of one of our first understandings of DNA, is actually just looking at a different angle from DNA, but you can see the stuff in the center, the helical pattern, sugar phosphate backbones around the outside. So why is there some controversy? Well, for many different reasons that's suggested, people call her the wronged heroine, the Dark Lady of DNA, because she actually didn't get credit for the discovery of DNA. It's going to come to two people that we're going to talk about soon, Watson and Crick. Some people say it was because she was a woman in the field. Some people say that she was the Dark Lady of DNA because her research wasn't taken seriously by all the men. And there's actually a lot of evidence to support that. So she doesn't really get the credit, and there's a lot of movement to give her a sort of a posthumous Nobel Prize. But the Nobel Prize didn't go to her. She also died pretty young, and instead went to these two folks here, Watson and Crick, who published their work on the double helix model of DNA. This one's Crick here, and there's Watson. Crick has passed away. Watson, I believe, is still alive here in 2021. But they developed this double helix model, right? The idea of two strands of DNA wrapped in this helical pattern, sugar phosphates in the backbone, and the bases in the center. So they got the Nobel Prize along with a colleague, Maurice Wilkins, but these two are generally credited with this discovery of the double helix model of DNA. Although we do know that controversy surrounds Rosalind Franklin, who probably should have been given the credit as well. Okay, so we talked about the discoveries that led to DNA. Now, what does modern science tell us? What can we really understand about DNA? Well, we know that DNA actually has a direction, right? There's some direction in DNA. So if we take a look here at some DNA, right? Remember, we got two strands. Here's one strand. Here's the other strand over here. We got sugar phosphate. So we got sugar phosphate, sugar phosphate. So that's a sugar phosphate backbone, we like to say. Sugar phosphate backbone. Oh, kind of moving all over the place there. And then in the center, of course, are the nitrogenous bases, of which there are four, A, T, C, and G. But if you take one of these nucleotides and sort of bring it over here, which is what I'm going to do, The direction of DNA depends on the end that's exposed. So we count these from the carbon of the sugar. So here's the sugar, right? That's our sugar. It's our ribose sugar for RNA and our deoxyribose sugar in DNA. So this would be carbon 1, 2, 3, 4, 5. So the phosphate group, notice, is attached to the 5th prime carbon. And if you notice down here, there is a sugar. This is the 3rd carbon that's on this end. Up here, notice that the 5 prime carbon, the one here, is the one on this end. And so we say DNA has direction. It has a 3 prime end and a 5 prime end. It's whatever one is exposed. Notice on this side, the 3 prime end is the one that's exposed, and down here, because there's a phosphate, the 5 prime end is the one that's exposed. So DNA has direction. Let's also talk about this DNA backbone a little more specifically. So I mentioned that DNA is made up of these nucleotides, right? There's a nucleotide there. And these nucleotides have 3 components: the sugar, the phosphate, and the base. So in DNA, what we see here, this sugar is a deoxyribose. It's missing an oxygen. RNA, which we'll see later, has a ribose sugar. There's also a phosphate, and then these nitrogenous bases are in the center. Notice here I've got the direction labeled because down here the 3 prime end is what's exposed. Up here the 5 prime end is exposed, so this is the 3 to 5 direction there. Over here it's opposite. The 5 prime phosphate's exposed there, and up here, notice up here we got the 1, 2, 3 carbon prime end that is exposed there as well. These bases in the center are really what determine our genes. It's the order of these bases that make up our different genetic traits. So this is DNA. RNA is generally a single strand, and we'll go over RNA a bit more in a different podcast. But RNA is often single-stranded, most of the time it is. And it contains a uracil instead of the thymine in DNA. And that is what separates RNA a little bit from DNA. Now, I kind of alluded to this, but DNA is in an anti-parallel strand configuration. So if this side over here, in this case it happens to be the left side, notice that it's running from here 5 down to 3. But this one over here is anti-parallel. So it's parallel to it, but it's in the opposite direction. Notice this one is 3 to 5. So because DNA has this direction, the strands are complementary to each other but anti-parallel. They run in the opposite direction. This is going to be very important when we talk about how DNA replicates, because these directions do matter, right? If you're just, I have a strand of DNA here, right, a little model. DNA you got to think of is it's 3D and it moves around, right? So at any point, if we want to say, well, what's the direction? We have to say, okay, let's hold it straight. And then we'll say, okay, here's the 3 prime end, here's the 5 prime end, so that we know the order of the bases. So it's important that we have a direction so we know what order these bases are in. So speaking of the bases, I want to talk about those 4 bases in DNA, and then there's that 5th one in RNA. So bases, these nitrogenous bases, we have some called purines, we have some called pyrimidines, and these will pair together in a certain fashion. So let me give you some more information about that. A purine is the fancy chemical word we use for these bases that have a double ring, and that's adenine and guanine. So if you notice here, guanine has 1, 2 rings. There's 1 ring, 2nd ring. Adenine also has 1, 2 rings as well. 1, 2. So guanine and adenine are the purines. The single ring bases are called pyrimidines. So thymine just has 1 ring. Cytosine also only has 1 ring as well. Now, a purine has to bond with a pyrimidine. So if you notice in these diagrams, cytosine always bonds with guanine. In other words, they pair together. they pair together with these hydrogen bonds in the center. And that's important because hydrogen bonds are actually pretty weak bonds, and that means later on these bonds can break because the DNA will open. Notice down here, thymine pairs with adenine. So the pairing is always a pyrimidine and a purine, and the adenine always pairs with the thymine, and the cytosine always pairs with the guanine. So A goes with T, C goes with G. Notice these two, thymine and adenine, they're only connected by 2 hydrogen bonds, but notice cytosine and guanine are actually connected by 3 hydrogen bonds in the center. So that's the base pairing. That's how these bases pair together. So if we go back to my model here, the bases in the center of this model pair together. So adenine goes with thymine, cytosine goes with guanine. Okay, so what does DNA do? We've talked about the structure, we've talked about the discovery of DNA. What does it actually do? Well, we pretty much have a good understanding that DNA stores our heritable information. It's what's involved in heredity. So genetic information is stored and passed to the next generation through this DNA. Sometimes RNA, but that's rare. Now, everything, all living cells, all living things have some chromosomes or some form of DNA. Our chromosomes, eukaryotes, we have linear chromosomes, whereas prokaryotes have circular. So here you can see typical, right, eukaryotic chromosomes there. This is a bacteria, so you can see inside that is a prokaryotic chromosome. In there is kind of circular. Okay, so eukaryotic chromosomes are often linear, prokaryotic chromosome circular. Now, this actually presents us with some really interesting things that we can do in terms of biotechnology, because we know that prokaryotes and viruses often contain plasmids. Plasmids are small, extra chromosomal, meaning they're not part of the actual chromosome, circular double-stranded DNA molecules. So I've got some examples here. This is a typical plasmid, and by the way, we can manufacture these, and we do this quite a bit in biotechnology. It's just a circular piece of DNA. And in this case, notice the bacterial DNA is already in there, right? That's the DNA for the bacteria. This right here is a plasmid that might get inserted. And the bacteria treats this like it's its own DNA, right? So it will do and express the genes and the traits that are in that little plasmid that we've inserted. So we could insert those into DNA. We can insert those plasmids into viruses as well. So to wrap things up, let's talk about the central dogma of biology, or the central dogma of molecular biology. If you think of like a dogma, that's just sort of like the main tenet. If you think of like a religion that has its dogma, sort of what it believes in, this is what is the central dogma of molecular biology, the flow of genetic information within a cell. So we've talked a lot about DNA. That's what it looks like. Occasionally, DNA has to make more DNA. We call that process replication. So in our next video, we'll actually talk about in detail how DNA replicates itself. Then DNA is also used as the code to make messenger RNA. And there you can see that single-stranded messenger RNA that's right there for us. It's basically a single-stranded copy of that. And then that process is called transcription. So going from DNA to messenger RNA is called transcription. Then we have messenger RNA ultimately being converted or used as a basic blueprint or a code to build our proteins. That process is called translation. Okay? So the central dogma, DNA makes more DNA, called replication. DNA used to make messenger RNA, called transcription. Messenger RNA used as code to make protein, that process is called translation. So hopefully this video helped you understand the discoveries that led to DNA, as well as some of the basic structure and function of the DNA that's within all living things.